let me introduce the second abstract presenter. Cheryl Pikora will present the challenges of Remdesivir Pediatric development for SARS-CoV-2 infection in a pandemic, and she's doing this presentation for Gilead. You have the floor, Cheryl.
I'd like to thank the meeting organizers for inviting Gilead to present on Remdesivir challenges of pediatric development for SARS-CoV-2 infection during a pandemic. My disclosure is that I am an employee of Gilead Sciences. The first challenge we faced was, at the time the pediatric development plan for Remdesivir was needed, we knew little about disease severity or incidence in children. US, EU, and Chinese publications in the spring provided insight and demonstrated a fairly low incidence of COVID in children. Few children are noted to have severe disease, while in the middle of designing the pediatric study, cases of multi-system inflammatory disorder were being reported. So it was clear that at that point, some children had classic respiratory symptoms, whereas others did not. The point of this slide is to show that pediatric COVID-19 cases have been on the rise from a rate of 3% in April, when we were designing the study, to now a little over 10% in October. And although most children don't require hospitalization, as many as 7.5% with a proven case now are ill enough to be hospitalized. At this time, Remdesivir was being studied as a treatment for COVID-19 in adults. Remdesivir is a broad-spectrum antiviral drug with activity against coronaviruses with a low EC50. Additionally, data from SARS-CoV-2 inoculated rhesus macaques showed a reduction in clinical scores in monkeys treated with Remdesivir versus placebo, and a reduction in pathological changes in the lung. The next challenge was choosing an appropriate study design in terms of using a single-arm pharmacokinetic or PK and safety study versus a controlled study. What helped us decide were preliminary results, now made final, of the Act 1 study. This was a double-blind randomized placebo-controlled trial of Remdesivir in adults hospitalized with evidence of lower respiratory tract involvement, given Remdesivir or placebo for up to 10 days. The primary outcome was time to recovery. The results of the study showed a statistically significant shortened median recovery time for Remdesivir relative to placebo. So this proven efficacy in adults allowed us to move forward with a simple one-arm PK and safety study design for children. This choice of study design is one supported by the FDA's pediatric rule, in which pediatric extrapolation of efficacy from adults can be done using PK bridging if the course of the disease and the product's effects were similar in adults and pediatric patients. The third challenge was the choice of Remdesivir dose to use in the pediatric study. PBPK modeling is a physiologically based model that considers ontogeny and age-related changes in organ development and function, and was used to generate a dosing nomogram for pediatric administration of Remdesivir. The fourth challenge is how we could obtain sufficient PK data from children with stringent precautions for COVID for hospital staff and in children who are potentially critically ill, requiring intensive interventions. We did not feel comfortable with an intensive PK sampling plan in this setting, but went with sufficient sparse PK sampling to be able to measure the metabolites shown above to use in a robust population PK model of Remdesivir in children that would ultimately provide the study's primary PK endpoint. A fifth challenge is Remdesivir is not orally bioavailable, so needs to be administered IV and has different pH requirements for optimal solubility versus chemical stability. The formulation needed cyclodextrin to increase solubility, while the study of Remdesivir in children required minimizing cyclodextrin. We therefore chose to go with the lyophilized powder for solution to minimize the cyclodextrin exposures. Another challenge was that we needed to move quickly during a pandemic to put into place pediatric plans for Remdesivir in order to start the implementation of these plans. Gilead had several meetings with regulators in the US and EU, as well as external experts, to help with the pediatric development plan, as well as the study design. This approach was highly collaborative and led to unprecedented timing in PIP and PSP approvals and study startup. Once the basis of ongoing accumulation of pediatric clinical data from published observations as well as the adult data were collected and input from regulators was obtained, the study referred to as CARAVAN was designed. Key inclusion criteria are lab-proven SARS-CoV-2 infection and hospitalization and absence of renal or hepatic failure. All children over 2.5 kilograms and at least 14 days of age are enrolled in parallel and divided into cohorts above in the interest of spanning an adequate range of weight with an appropriate sample size. Participants are treated up to 10 days with daily remdesivir, and clinical assessments in multiple safety labs as well as PK assessments are performed as indicated on the slide, with a final visit at day 30. A significant challenge to this design was the size of some of these participants and the blood volume limits, for which we consulted a pediatric laboratory specialist. In conclusion, despite these challenges, we initiated the study successfully and enrolled our first participants in July. As of October 19, there were 20 participants enrolled across cohorts 1 through 4. We've had enrollment challenges as children seem to be managed more conservatively now, having learned from earlier cases, and fewer given a remdesivir course. The rate of symptomatic and hospitalized children has been fairly low, which necessitated opening additional sites. So we're over 25 sites at this point in the US and EU. We have an external data monitoring committee or IDMC meeting coming up soon to review the safety outcomes, which is triggered once 50% of subjects have been enrolled and reached day 10. And we reassess on a daily basis whether new sites might enhance enrollment based on epidemiologic data, and we anticipate study completion at the beginning of next year. Lastly, we like to express our solidarity with those who have been ill or are ill with COVID-19, their families, and the healthcare workers on the front lines of this pandemic. And we extend our thanks and appreciation to the CARAVAN study patients, their partners and families, the frontline healthcare workers caring for them, the study staff, and study investigators. Thank you very much.